Good evening everyone. Welcome back to my channel My Way of Learning. So today we are going to see the very important topic in NEET biology, that is the cells, the basic unit of life. So from this you can expect many of the questions in the NEET exam, and people who are preparing for TNPSC mains also the cell topic is coming. So please listen the video, like, comment and share and subscribe my channel. So for the further more videos, please press the bell icon also. Let's get into the video. So first the cell. So you all know what is a cell. So cell is a single unit which is together makes a similar cells will make a similar tissue, and a similar type of tissue makes a organ. So this is how our body is made up of. So there are different types of cell. First let's get into the introduction of cell. Who has discovered the cell for the first time? Robert Hooke in 1665 was the first to saw the cell in a thin slice of cork with this crude microscope. So the cork under microscope resembled like a honeycomb structure which is made up of several compartments. So Robert Hooke only called this compartment as cells. So as I said before, a tissue is a group of cells of similar structure and function arranged in the body, so as to give the highest possible efficiency to the function they perform. So there are lots of difference between a unicellular and a multicellular cell. So all living organisms are made up of cell. In a unicellular or acellular, unicellular also called as acellular organism, a single cell may constitute a whole organism, like example of amoeba, chlamydomonas, so many of the bacteria. So these are unicellular organisms, so in which the whole cell performs all the functions. The multicellular organisms are nothing but which constitute of many cells grouped together in a single body and assume different functions. Example, fungi, plants, animals, etc. So we all multicellular organisms. So many cells makes up a tissue, and tissue makes up organ, and thus it is assembled in a body, and which is doing a different function. Each cell is having different function in a particular organ. So that is how the multicellular organisms are living. And various cells of human body vary in structure according to their function also. So each living cell perform certain basic functions that are characteristic of all living forms. So from this you have to know what is a prokaryote. As I said, each of the living organism is made up of cell, which is the basic unit of the life. So what is a prokaryote? Prokaryote is nothing but a unicellular type of organisms. Example, bacteria and some other organism lack a cell wall and a definite nucleus surrounded by nuclear membrane, and membrane bound cell organelles also absent in the prokaryotes. So this type of organisms we call it as prokaryotes. So eukaryotes are nothing but the organism with a well defined nucleus surrounded by nuclear membrane bound cell organelles which are present in this eukaryote. So this is the main difference between the prokaryote and the eukaryote. I am repeating it again. Prokaryote which lack definite nucleus and surrounded by nuclear membrane, and membrane bound cell organelles are absent. And in a eukaryote it have a definite nucleus and well defined nucleus and surrounded by nuclear membrane, and membrane bound cell organelles are present. These are the major two difference between the prokaryote and the eukaryote. So those organisms which possess this which possess the defined nucleus and membrane bound cell organelles comes under eukaryotes, which doesn't possess comes under prokaryotes. Okay, so coming back to the structural organization of a cell. So how the cell has been made. Okay, so how the cell has been made, how the cell has been, what all the has been made, what are the structural organization of a cell. So the parts are plasma membrane, or also called as cell membrane. Okay, so this plasma membrane has been very important because it is the outermost covering of the cell, which separates the contents of the cell from its external environment. So how a unicellular organism survives in an environment with the help of the cell membrane, that is the plasma membrane. It allows entry and exit of only certain materials. So it is also called selectively permeable membrane. So the membrane which allows only a selective material inside and outside, that we call it as selectively permeable. So this type of membrane is our plasma membrane. So we call it as selectively permeable membrane. Likewise, the carbon dioxide and the oxygen moves across the membrane through diffusion. So diffusion process, the movement of the molecules from higher concentration to lower concentration, that is called as diffusion, right? So this plasma membrane structure has been proposed by Singer and Nicholson in 1972. That model we call it as the fluid mosaic model of plasma membrane. It is completely made up of lipids and proteins. Okay, this is a very important question when the facts about or the information related to a plasma membrane also they may ask. Okay, diffusion I have explained you. It is the movement of substance from a region of higher concentration to a region of lower concentration, right? Okay, so osmosis is nothing but the movement of water, especially through a selectively permeable membrane, from a region of high water concentration to the region of low water concentration. So diffusion is not restricted to any of the molecules. So any type of molecules will happen, but osmosis is completely related to the movement of water. We also call it as solvent. So the movement of solvent through a selectively permeable membrane from a region of high water concentration to a region of low water concentration, the process is called as osmosis. Okay, so the fate of animal or plant cell in a hypertonic and hypotonic and isotonic solution. So that is also very important when you are coming to learn about the plasma membrane. So plasma membrane is one which is selectively permeable, and it plays a major role in the exit and entry of various materials, right? So inside and outside the cell. So what happens when the cell has been kept in a hypertonic solution? So many questions has been asked based on this hypertonic, hypotonic and isotonic solutions. So please listen carefully. Hypertonic solution, the concentration of solution is more than that of cell. Okay, so solution is nothing but a liquid formed by mixing a solute and a solvent, right? So solute and solvent mix together and forms a new product that is called a solution. So when the concentration of this solution is more than that of the cell, therefore the cell will lose water by exoosmosis and it will shrink. So here the hypertonic denotes the concentration of the solution which is more than that of the cell. So the solution present inside the cell compared to the environment is less. Okay, so the environment concentration of the solution is more than that of the cell. So that time the cell will lose the water by the process called exo means exit, right? So it will losing the water and it becomes shrink. So like a water balloon you can think of for hypotonic solution. So when the water balloon loses the water, it shrinks, right, like that. Okay, so next one, hypotonic solution, the concentration of solution is less than that of cell. So here hypotonic means the concentration of solution in the environment is lesser than the concentration of the cell inside. Therefore, the cell will gain water by endoosmosis and swell up. So by gaining water, it will swell up. By losing water, it will shrink. This is a very basic process. We are seeing it in a daily life, even in a vegetable also. So you can easily understand this. Okay. Next one, what is isotonic solution? The concentration of solution is same as that of the cell and the outside environment. Both are in a equilibrium. So that is called as iso. Iso means same. Hypo means less, hyper means more. So here hypertonic, more concentration outside environment, less concentration inside cell. Hypotonic solution, less concentration outside environment than the cell, more concentration inside the cell. But in isotonic, both concentration are equal. So therefore there is no movement of water and cell size will remain the same. So the questions may be asked like that. What happens in the hypotonic solution? By the process of exosmosis, the cell shrinks. By when the cell is kept in hypotonic solution, the cell will gain water through endoosmosis process and it will swell up. And in isotonic solution, no water movement is happening and the cell size remains the same. So these are very important related to the plasma membrane, that is the cell membrane of a cell. Okay, so the next one is cell wall. Okay, so the cell wall is very important for organisms, right? So it is the outermost covering which lies outside the plasma membrane. We have seen the plasma membrane as the outermost covering of the cell. Above the plasma membrane lies the cell wall. So plant cell wall is composed of a complex substance called cellulose, which provides the structural strength to the plants. Fine. So this is very important because the cell wall is present in plant cell, not in animal cell, right? The plant cell needs a very structural strength. Rigidity is more important than compared to the plant cell. Okay. Because it is directly related to the outer environment and it has to withstand all type of weathers. So the cell wall is very important. So it is made up of the substance called cellulose, which provide the structural strength to the plant. Due to cell wall, plants, fungi and bacteria withstand much greater changes in the surrounding medium than animal cell. So the bacteria coming under the prokaryote lacks definite cell wall, cell nucleus. Okay, but only by the nuclear membrane it is surrounded. It doesn't have a well defined nucleus, but also it doesn't contain a membrane bound cell organelles. So but it contains a cell wall, so which make them to withstand the surrounding medium than the animal cell. Okay, so plasmolysis, what is mean by plasmolysis? The shrinking of protoplasm of a cell due to exoosmosis when kept in hypotonic solution. So plasmolysis, when it happens, the plasma membrane which is holding the all content of the cell and protecting the cell from the outer separating the content of the cell from the external environment, right? So when it kept under the hypertonic solution, due to exoosmosis process, it will lose the cell content, right? So that type of losing leads to the shrinking of protoplasm, and that we term as plasmolysis. Okay, so it will the functions of this plasmolysis is to exhibit a plasmolysis process when kept under a hypotonic solution. Okay, so coming back to the functions of cell wall. Cell wall, it provides rigidity and the strength to the cell, that you know. It also helps to sustain during unfavorable conditions. So these two are the main functions of the cell wall. This I have said already. Okay, the next structure of the cell is nucleus. Okay, so what do you think that all students need to have an understanding of the parts of a cell and the difference between the part found in plant and animal cell? Just now I am explaining the parts as shown in the slide. So please listen carefully. Okay, so here what we are doing is which is the site of ribosome formation and released out into the cytoplasm and reaches this rough endoplasmic reticulum and makes the endoplasmic reticulum into a rough by attaching to its surface. So here the ribosome serves as the site of protein synthesis. So the smooth endoplasmic reticulum, it helps in the manufacture of fat and lipids. So I repeat again, vacuoles are large in plants and small in animals, and it functions as the storage sac for solid or liquid contents, and it provides turgidity and rigidity to the cell, and the contractile vacuoles helps in expelling excess water and waste in some animal cells. So the next one, placid, I said they present only in plant cell. There are of two types, the chromoplast, which is the colored plastids, and the leucoplast, which is called as white plastid, colorless. The plastid that contain green color pigment is known as chlorophyll. The green color pigment chlorophyll is known as chloroplast, and they have their own DNA and ribosome. Likewise, the mitochondria, double membrane bound organelles, which is porous, while the inner membrane, outer is porous and the inner membrane is deeply folded, large surface, providing large surface for the ATP generating chemical reaction. It is also called as the powerhouse of the cell. This mitochondria also have the own DNA and ribosomes. And the lysosomes, these are membrane bound sac filled with digestive enzymes. When cell get damaged, lysosome burst and enzyme digest their own cell, so they are called as suicide bags of the cell. So it keeps on cleaning the cell by digesting foreign material and old worn out cell organelles. Okay, so the Golgi apparatus discovered by Camillo Golgi, and it consists of membrane bound vesicles arranged parallel to each other in stacks called cristae, connected with endoplasmic reticulum, and it helps in transport of substances synthesized near endoplasmic reticulum inside and outside the cell. It helps in storing, modifying and packaging of products in vesicle. So this is all up to the structural organization of a cell and the parts of the cell. The main organelles I have explained. For further one-to-one class, you can contact me. I will be sharing my mail ID. You can message me and get my contact. And we are offering this NEET biology for all at a very low cost. So one-to-one class will be more elaborated with practice session of questions under each topic. Likewise, for the TNPSC science related topics, general science related topics can be also taken one-to-one. So please contact us for further details. Thank you all. I'll meet you with the next topic, that is the tissue. So please comment, share and subscribe my channel. Thank you.